Hey, BioTeam. So let's say that there's a chromosome that has a gene on it that our body wants to read and code into a protein. Well, the first thing that our body would have to do in order to access that gene on the chromosome is it would need to unzip the chromosome. That is, it would have to take these two strands of DNA and somehow separate them apart. And this process is harder than it looks because the strands of DNA are attached to each other. That is, if we were to zoom in on the base pairs, what you'd see is that every base pair is made up of a one-ring base called a pyrimidine and a two-ring base called a purine. And you'd see that these base pairs are connected to each other via hydrogen bond. And so you'd see that every adenine and thymine is held together by two hydrogen bonds. And you'd see that every guanine and cytosine pair is held together by three hydrogen bonds. And so what our body will do if it wants to get at the code inside of a helix of DNA is it will use an enzyme called DNA helicase, which is the enzyme that's responsible for breaking apart these hydrogen bonds. And what DNA helicase will do is it will look for what's called a TATA box, which is just a group in the helix of adenine and thymines clustered together. And the reason why DNA helicase will look for a TATA box is because a TATA box is really easy to break apart because these adenine-thymine pairs have fewer hydrogen bonds holding them together than guanine and cytosine. And so if helicase is looking for a good place to start breaking apart the chromosome, it'll find these weaker base pairs. And so what helicase will do is it will literally come in between the adenine and thymine and wedge those two apart. It'll move in between these base pairs and start splitting apart those hydrogen bonds. So how does our cell read a gene after it's been unzipped? Well, if you were to zoom in on nucleotides, what you'd see is that the sugar in each nucleotide has 5 carbons. And so there's 2 ends to each nucleotide. There is the 3-carbon end of the molecule, which connects to the next nucleotide in the strand. And then there's the 5-carbon end of the sugar, which connects to the phosphate. And so this entire strand of DNA, in this case, has a 3 end to it, where the 3-carbon is, and it has a 5 end to the molecule, where the 5-carbon is on this nucleotide. And so when it comes time to read this strand of DNA, nucleic acids are always read from the 3 end to the 5 end. And that's it. At this point, you guys have some practice problems. We'll see you next class.